Okay, so welcome back everyone. And today's lecture is going to be on stem cell research and vaccines that are bioethically controversial. Not all are, but some vaccines are because of the way they were developed. So I'll get to that topic in the second part, but the first part is dedicated to stem cell research. Okay, so as always, we begin with a little prayer. Come, Blessed Trinity, to fill our hearts and to be with us. Help us to discern these issues in bioethics on healthcare that can be complicated at times, but with your grace, we can come to an understanding and to respect the dignity and the sanctity of human life at every stage of development, and at the same time allow for the progress and development of a good medical practice. As always, we pray for those who are in most need of your divine mercy, in the name of Christ our Lord. Amen. All right, any questions or comments from previous stuff? No? Okay, so we're going to move forward then. First, stem cell research. When we ask, for example, just a simple question like, do you think that the Catholic Church is in favor or against stem cell research? What's your intuition there? Against, right? But actually the way the, what is, you Anna, what do you think? For or against?
I thought it was
for. Okay. Actually, the way I framed the question, the proper answer would be, it depends. Exactly. We have to make some distinctions. That's the thing. And remember, forever in bioethics, we are making distinctions. You know, what do you mean by stem cell research? What kind? So we have to make a distinction at least at two levels. At the level of human and non-human research, okay, because of course we assume it's human research, but there's non-human stem cell research going on. In fact, the bulk of the stem cell research is non-human right now. And then there is the distinction between adult or embryonic stem cells, all right? And so those are the two levels of distinctions that we have to make before we can pass an ethical judgment. And that's why basically we always start with the basic biology, all right? So moving forward then, we're going to look at the first level of distinction, which is between human and non-human. And what do we mean by that? So,
animals and humans, the difference between.
Yes, exactly. So there is a type of research that is being done in experimental animals, mammals, and even non-mammals, but mammals is the preferred model for eventually translated into something clinical for humans. All right, so that's the first level. We're talking about stem cell research in mice, for example, or we're talking about the research in humans. All right, so let's say that it's a human stem cell research. We still need to make a second clarification before we pass an ethical judgment, whether it's going to be adult stem cell or embryonic stem cell, okay? So that's the second level. Because bioethically, it makes a difference. Again, we'll look at the biology first, and then we'll see why it makes a difference between embryonic and adult stem cell. And basically, the punchline is this. Where do the embryonic stem cells come from? What is the source of the embryonic stem cells? Sorry? Well, it's a little beyond the zygote. Zygote is the first cell, all right? But it is the early embryo. What stage? We have the zygote, from zygote to morula, right? And then after the morula comes blastocyst, exactly. The blastocyst, right? The blastocyst, exactly. Before gastrulation, in other words, before implantation. We have to understand that when we talk about embryonic stem cell research, human and even non-human, it's not that the researchers are going into women's wombs and pulling out embryos, right? They're not implanted, that's the whole thing. But it is the inner cell mass of the blastocyst. So if you remember when we talked about the biology, the basic biology of early embryonic development, right, up to gastrulation, remember? Then at the blastocyst stage is the first level of cell differentiation. Remember when we have two cell layers, two distinct cell layers, which are, you remember? Trophoblasts or trophectoderm, which is the outer layer, right? And then inside, exactly, inner cell mass known as ICM, okay? And it is actually the ICM that is used for generating these embryonic stem cells. It is actually the ICM, right, the inner cell mass. And why is that?
Because they're not so important.
Well, pluripotent, right, and existentially, functionally, what does that mean? Remember, I said that little phrase, the embryo proper comes from the ICM. In other words, it's the ICM that develops into the embryo because it's the ICM that differentiates into the three germ layers, right? Ecto, meso, and endoderm, and the gastrula. So it's the ICM, the gold standard, if you will, because in principle, all of the organs and tissues come from the ICM, right? And therefore, they're the ones that have the greatest potential to heal all the organs and tissues that are damaged. Mm-hmm. See how it works? So that's what's known as the gold standard. But it's actually a theoretical gold standard. In practice, it's not that good, really, and you'll see why. And this is strictly on the biology of it, all right? Even before we do the bioethical analysis. But basically, the embryonic stem cells, which are known as ESCs, right? ESC is we're kind of we're fixated on acronyms, right? We always want to make acronyms for significant little phrases. So the ESCs are the embryonic stem cells. They are the ICM. They are the inner cell mass, which is disaggregated. It is plucked out of the blastocyst and placed in a petri dish or some kind of a culture medium that culture medium is going to mimic the internal environment of the reproductive tract, because that's the native environment for those cells in which they thrive, right? Okay, so in this aggregating, in isolating the ICM from the rest of the blastocyst, what happens to that blastocyst? In effect, it dies, but it's killed, right? The blastocyst is actually killed when the ICM is disaggregated. Jody, welcome. All right. Okay. Again, you're welcome. And therefore, that's the issue right there. That's the bioethical issue, that in order to obtain embryonic stem cells, the embryo, the blastocyst, the early embryo, has to be killed. All right, has to be killed. So that's the issue right there. Okay. Now, with regards to whether they're human or not human, it makes a difference, because whereas we may justify the killing of mouse embryos if by that killing we're able to obtain some results, can be translated into a clinical benefit, right, to some kind of healing benefit in the experimentation that is done in the research, then the killing of a mouse embryo, just like the killing of an adult embryo, of an adult mouse, can be justified bioethically if there is some benefit to be obtained, right? And the protocols are pretty stringent, by the way. They have to go through a review board and so forth to make sure that the animal is euthanized in a humane way and so forth. It's different when we're talking about human beings, because what is the basic necessity when experimenting with human beings? When the subject of experimentation is a human being, what is the basic requirement, non-negotiable?
Total brain death.
Well, then we're talking about a cadaver. You're still on transplants. Consent, voluntary, that the person has to consent to this research. So we can be subjects of research, we can volunteer into clinical trials. They exist because they have to test the drugs at some point in some humans, all right? But that consent needs to be informed, for example. In other words, I need to know what kind of risk I'm going to sustain to go into this research. And then I decide yes or no. Okay, if the risk is proportionate and so forth, if there's going to be some benefit and the risk is minimal, fine. But that consent, how do we obtain consent from a human embryo? Impossible. Okay, and so we say, well, what about the mother giving consent? First of all, these are not implanted embryos. So they're actually in a lab. Typically, they come from in vitro clinics, all right? The spare embryos that we talked about in the tanks, in the cryopreserved tanks. So what if the parents give consent? Yeah, but they're consenting to kill their child. So it's not, it's not logical, or it's not ethical to consent to the killing of another human being without their consent. Okay, and so that even if we say, well, it's proxy consent, we cannot go that far because the proxy has to be assigned by the person before they fall into a coma or before they become discapacitated, right? So that's the whole issue here. There's no way that the embryo can consent to this. Plus, it's research that is lethal. It's lethal for the embryo. So that's the issue there. If there was a way that we could obtain embryonic stem cells without damaging the embryo significantly, then we would actually be in favor of it. In fact, just asking the question like that, is a Catholic Church for or against stem cell research? Many people will say at first no, and I just like to be a little controversial, say, well, actually, just framed that way, the church is actually involved in stem cell research. It's not against, but actually involved in stem cell research. But of course, once we do the clarification, it turns out that it's going to be adult stem cell research, which is the one that is actually working today. Okay. So before I move forward then, you're clear on the source of embryonic stem cells, right? It's the human embryo by definition. And that human embryo has to be disaggregated, has to be killed, all right? They talked about destroyed. This is the euphemism of language. The embryo is destroyed. And that's where, again, we have to be very sensitive to these issues if you're going to be in healthcare, because you'll hear me say language is significant. Words have meanings. What do we destroy normally? Okay, if we were to destroy something, what is it that we destroy? Well, we destroy an old car, we destroy a chair that's falling apart, like we recycle it, would be a way of destroying something. A desk is falling apart, we destroy it, we recycle the wood, all right? But basically, we destroy objects. We don't destroy people. Like if I say I destroy another human being, it sounds pretty nasty. Destroying a human being is actually killing the human being. But they don't want to say killing the blastocyst, killing the embryo, because then we are humanizing the embryo. And this group or the people who are intent on doing this research, of course, they want to dehumanize the embryo. They don't want to make the embryo seem human for what he or she is, because then the ethical issues can crop up. Okay, so they use this language of destroying. They'll talk about destroying the blastocyst, trying to make it as objective, as unhuman as possible. Okay, they won't talk about killing the early embryo, for example. So as professionals, be very attentive to the type of wording that is being used, because you'll find this in the field. You can't expect everyone to have the same value system that we have, right? And believe me, there's a lot of money. There's big money in this, billions of dollars, because this has potential for curing cancer and diabetes and neurological diseases and trauma. Okay, so there's potential for big healing here. Okay, now let's look at potency for a moment. It's like a little parenthesis in between because a lot has to do with potential, all right? The potency of the cells. Again, why is the ICM the gold standard? In terms of potency, the ICM is pluripotent, okay? What does pluripotent mean?
It has, it can discern from the differentiation.
Any organ or tissue of the body. The 220 lines that I've talked about, the cell lines that make up our different organs and systems, okay? It's approximately 220 cell lines. And that's the whole issue with the embryonic stem cells. They want to develop at least the precursors of these 200 plus cell lines to eventually be able to replace every organ or tissue that is injured or diseased in the human body. It's a very high goal, right? But that's the whole issue of pluripotent, all right? Now, they're one step removed from totipotent, right? What are the totipotent cells in the human body, in the human organism? Give you a couple of photographs here, microphotographs. What is this? This is zygote, right? Just before syngamy. You see the two haploid pronuclei here. Okay, these photos are in the internet, and in fact, this is the Sunna Pellucida and the corona radiata is still around it. This is the fertilized state with the two haploid pronuclei about to fuse and restore diploidy. The cell as a whole is already diploid, is the zygote, but at the gametic level, the gametes have not fused yet. Okay, and then what's here underneath? Morula, right? Morula, see, little berry with all the blastomeres. Remember, each cell is called a blastomere. Okay, so these are totipotent. Obviously, the zygote is totipotent because that's the only cell there is, and so the whole organism comes from the zygote, regardless of what organism, whether it's animal or plant. Any species that reproduces sexually, the first cell is going to be a zygote, right? By definition, the link or the yoked a cell of fusion between egg and sperm. So zygote and morula are totipotent, right? By the time we get to the blastocyst, because there has been differentiation, the first level of differentiation, these cells, these individual cells, which are still called blastomeres, all right, are no longer totipotent. So it's unfortunate that both the cells of the blastocyst and the morula are called blastomeres. Why is unfortunate? Because the blastomeres of the blastocyst don't have the same potential as the blastomeres of the morula. Okay, the morula blastomeres are totipotent, so that if a morula is disaggregated, each one of those cells, each one of those blastomeres, has the potential to become a new zygote, a new individual. But that capacity is already lost at the blastocyst stage, because there has been differentiation, the first level of differentiation. Okay? You with me? Okay. All right, so these are pluripotent, and the gastrula, all right, the gastrula is also pluripotent, which is once that ICM continuous differentiation into the 3 germ layers. What are the 3 germ layers? Ecto, meso, and endoderm, right? So that the ectoderm is not the mesoderm, and the mesoderm is not the endoderm, and different organs and tissues are going to come from those 3 layers. So there's been a further level of differentiation, but in general, because all of the organs and systems come from those 3 germ layers as a whole, maintaining the gastrula intact as a single individual, then they are also pluripotent, right? Those cells are pluripotent because globally, looking at the gastrula as a whole, right, the 3 germ layers exist within that one individual. So those are considered pluripotent. So you can see that there's a range of pluripotency. There's no range of totipotency. Cells, when cells are pluripotent, then they can generate a new individual. End of story. So there's no gradient, there's no range of pluripotency. It's either pluripotent or not. But for pluripotent, there's a range. So that the blastocyst is more pluripotent and the gastrula is less pluripotent, you see? But still pluripotent, because in general, all of the organs and systems are going to come out of there. By the time we get to birth, and even before birth, I would say beyond, you could probably make the case beyond where? When we have pretty much the organ systems in place, even though in miniature. What does that mean? When does that happen? Again, in the human embryonic development. We talked about this. The change in word is symbolic from embryo to fetus. When? Approximately 8 weeks or 2 months, right? Or pregnancy. What happens there? Very much the embryo within 8 weeks approximately will have all of the organs and systems formed in place, but in miniature, in place generally, okay, in the same general region of the body, head to tail. But in miniature, because that whole embryo is about how long? About 1 inch, all right? So we're talking really miniature. From that stage, for the next 7 months, it's mostly growth in size, you see? But the organs are basically developed. They are basically differentiated into the different organ systems so that the lungs will not become heart, and the heart will not become liver, and the liver will not become brain, etc., etc. So at that stage, we go from pluripotent to multipotent cells. And multipotent cells are within a particular organ system, okay? Multipotent are within an organ system. It could be the circulatory system, the dozen systems that we have approximately. So we're talking about the nervous system, we're talking about the respiratory system, circulatory, digestive, etc. See? And each one of those systems has come from a different germ layer to begin with. So there's a gradual loss of potency, right? With an increase, what? An increased differentiation. The cells, as the cells become differentiated, which is mostly embryonic development, right? At the cellular level, it's differentiation, becoming fully differentiated into particular organs and tissues. There is potency loss, so that they get channeled into a particular function, right? It's all about the function and not this other function. Okay, so beyond the 2nd month, approximately, in general, for most systems, the cells are becoming multipotent until eventually the vast majority of the cells in our body are fully differentiated, okay? And that will be the 4th stage of potency, fully differentiated cells, as I show here with the example of a neuron. But we still retain some stem cells, even in the adult stage, all right? For example, if I get a cut, I sustain a cut, what's going to happen to that cut if I keep it clean and just kind of leave it alone? It will heal. And does it heal? For the 2 options, from the outside in or from the inside out? From the inside out. It's nothing external, it's internally. In fact, if the cut is kind of deep and wide, as it heals, you'll see eventually, first you can see the layers of tissue, right? Down to the conjunctive tissue or whatever. But then eventually you'll see a pinkish layer of skin coming up. And that is the dermal layer. And eventually you get the epidermal layer, all right? And even the dead cells, so we have a whole layer of dead cells, which is beneficial because it's part of the insulation and parts of keeping bacteria out of the body and so forth. Oops. Yes, so you see that where does that skin, the new skin, come from? The new skin comes from stem cells and precursor cells that are deep, mostly in the adipose tissue and other conjunctive tissue in the lower layers of the dermis, of the skin, all right, of the integumentary system. So we have stem cells in our body, in different parts of the body. The liver, I mentioned, regenerates quite well, has a lot of stem cells. Different organs and tissues have stem cells in them, but they tend to be few in number and kind of hidden, all right? They don't like to be exposed, but they have found stem cells in adipose tissue, in the pulp of baby teeth, for example, as we shed the teeth, the pulp of baby teeth, hair follicles that grow new hairs and so forth. Also in adipose tissue, I mentioned that, right? So people can say the liposuction for stem cells. That's tongue-in-cheek. But there are an increasing number of adult stem cells that have been found. Oh, yeah, in the blood system, there have been found stem cells just traveling in the blood system. Again, they're few and kind of spread out, but they're there, okay? So that's exciting when they can be cultured in the lab and coaxed to grow and to do confluence in petri dishes and so forth, like we do with cell cultures. All right, so the adult stem cells are called ASCs. This is the acronym, adult stem cell, okay? And the first thing I want to point out is that this adult is really should be in quotations, in the sense that who qualifies as an adult as far as stem cells? A newborn. In other words, the non-embryonic, okay? Once one is out of the womb, then technically one has adult stem cells. So, for example, we adults, we have stem cells. How about teenagers? Yeah, they have stem cells too, okay? How about children? They have stem cells. Keep going back. Babies, newborns, they also have stem cells, all right? Now, they're not legal adults, all right, or chronological adults, but as far as stem cells, these adult stem cells are from anyone who is born. So what I'm trying to say is, and one could obtain stem cells, let's say, from the fat tissue of a baby without major risk to the life or even to the health of the baby, right? And the parents could possibly consent to that if those stem cells could serve for healing a sibling, for example, of that baby, which would have a good compatibility. Talk about transplants, all right? That would be stem cell transplant from one sibling to another. And that's a good thing, and the baby is not going to be harmed significantly by obtaining some of his or her fat tissue, right? So you see, that's a case where, and the baby then technically is or is contributing adult stem cells, even though chronologically, of course, a baby is not an adult, all right? So this adult stem cells applies to anyone born. But still, the proper consent has to be in place, and we have to be thinking all the time. We have to use our brains to see, well, is this really damaged the child or the teenager significantly, or is there a great benefit to be obtained with some kind of a minimal discomfort, all right? But certainly not the life of the child, less in the case of the embryonic stem cells. You follow? So the proportionality here, it can be justified. All right, typically adult stem cells are not pluripotent, they're multipotent, right? Why?
Because the organ system is already fully...
Already, exactly. We have mostly fully differentiated cells, fully differentiated organ systems, and so we're looking for those pristine cells, those precursor cells that are still around, okay? But we have gone beyond the pluripotent stage. And this is actually an advantage. You know that I said that it was a disadvantage for the embryonic stem cells to be pluripotent, even though they are the theoretical gold standard, theoretical, but this is a case where theory doesn't match the practice. Why are these embryonic stem cells not ideal in practice? Exactly. They actually have too much potential. They have too much potential. So in practice means functionally in the lab, when these experiments have been done in mice, precisely in embryonic mice, taking the ICM of mice and culturing them and then transferring those embryonic stem cells into the injury of the same mouse, so it's autologous, or, well, couldn't be the same mouse, but it could be the mother of the embryo, for example, which will have, let's say, 50% compatibility, or the father. What they do is they generate an injury on the mouse itself, right? Typically, the one that's best known is the spinal cord, for spinal cord injuries, because neuronal cells are the most specialized and fully differentiated, and ones that are more difficult to generate generally, right? So they take from the embryo the ICM, culture the stem cells, and then transplant them right into the area of injury, all right? Typically, what happens? There are two issues that arise. One issue that we talked about in transplantation, those embryonic stem cells, what's the compatibility with the mom of that embryo or the father of that embryo? What's the compatibility? 50%, right? 50%, so it's ambiguous. Therefore, there is the possibility of rejection, right, immune rejection, and therefore that mouse has to be put on suppressants, all right? The same would be for a patient, for example, for a human patient with embryonic stem cells. Necessarily, they don't come from the same patient, and therefore the patient has to be put on immunosuppressant. That's the first challenge. We can surpass that challenge because that's done with transplantation, you know, and people who are immunosuppressed can live a normal life. Just be careful in their lifestyle. But what's the biggest issue is precisely that typically when this is done experimentally, it has been shown time and again that those embryonic stem cells, instead of healing the injury, they turn into cancer cells. They grow uncontrollably. They grow uncontrollably, which is the core of cancer growth, right? It's uncontrolled. They don't know when to stop. So they actually grow a tumor there where they're supposed to heal a site of injury. They grow a tumor because they don't know when to stop. And that's why I'm saying that pluripotent is too much potential, too much potential. I give a simple little analogy. If you have a radio, when the radios used to be transistor radios with batteries, well, you can still find them. But that battery is typically what? It's a 1.5 volt battery, or even a 9 volt battery, you know, the square batteries. What is the voltage? 9 volt maximum, right? So now I have this transistor radio, but I have one of those 9 volt batteries, but instead of 9 volts, it has 200 volts. It's a supercharged 200 volt battery. When I put that battery into the radio, it's going to fry the radio. You know, it's too much potential, and that's the whole point.
So that's only if the adult stems, if it was pluripotent, it would cause it to grow the tumor cell.
Right, exactly. So this is a case where functionally it's better to be multipotent than pluripotent, because the pluripotent just have too much potential and they just don't know where to stop. It's like a wild horse. It doesn't know when to stop on the race, okay? So the horse has to be trained, and that's why multipotent are already predetermined in that particular organ system. So you will use stem cells in the blood for circulatory healing. You know, you use stem cells in the muscular system to heal muscle tissue or bone tissue, which have the same source, and so forth. So you want them to be multipotent because they're already predisposed for that particular organ system that what is seeking to heal, all right? And that's another advantage of the adult stem cells, is they're multipotent, they're toned down. They're not as strong and as wild as the pluripotent. They can also be autologous, meaning from the same patient. And this, again, is a case. This is a classical case of the autograft, right? Is stem cells from one part of the body that are used in a different part of the body of the same patient. No immune rejection, right? So that's very sweet. And in fact, there have been many, many successes obtained with these adult stem cells at different stages of development. And I'll give you a website here that is called stemcellresearch.org. Has been going on for quite a few years now. And this, you just punch in stem cell research as one word, and it should pop up. Let's see. Beautiful. There we go. It's a website that was created quite a few years ago by a combination of physicians and bioethicists, and it gives you, they have been logging all of the successes with adult stem cells and the lack of success from embryonic stem cells. And to this day, they have recorded 73, okay, they keep a track here, 73 successes in different stages of development and different organ systems for the adult, and so far 0 embryonic. One can go here somewhere. And it's got the papers. What they do is, once a paper appears, an article appears, they classify them into all these different categories, all right? And they have been maintaining, manicuring this site for, I think, about 10, 15 years now. It's a group called Do No Harm. And I think they're out of Washington, D.C. or somewhere, but let's see. Yeah, they're in Washington. Okay. Anyway, it's a very positive site because they have all this documented and recorded at the scientific level with the journals. And so far, nothing from the embryonic. After many years of animal trials and seeing the typical result of a cancer, you know, FDA, right? FDA, which is, what is FDA? Right, Food and Drug Administration. Yeah, Food and Drug Administration. You're thinking of...
Close,
yeah, right. You're thinking of what's the other one for agriculture?
Yeah,
It'll come to me. But anyway, FDA is the one that regulates, that gives the permits for doing human trials, clinical trials. All right, and finally, a few years ago, there was, I think it was Germ Corporation, which was one of the first biotechs that appeared in the 70s or 80s out in California. They were able to put together a protocol for something that was non-vital, something, I think it was a macular regeneration or something like that, okay? And so certainly it was not a vital function of the human body, but they managed to put a trial together for embryonic, for human embryonic stem cell research, because there was Proposition 71 many years ago that in California carved out about $3 billion from taxpayers to do embryonic stem cell research, human embryonic stem cell research at the state level before actually the federal funds opened up for that. And so they were pressed for putting together a human clinical trial that would justify all that huge expense of billions of dollars in the research. Well, Germ was able to put a protocol through FDA and for regeneration of macular degeneration. Within a few months, they pulled the license and they rescinded the research because they started coming up with issues in the pre-trials that it was showing that it was going to lead to tumors, to generation of tumors in people. So just as they were getting ready to go to the actual clinical trials, in the pre-trial testing, the tumors came up again. And so they pulled the license before they got sued for a few $1 million and shut that down. Okay, so to date, nothing. Even though they've tried and tried with all kinds of money pumped into this area, nothing has come up, which is providential because the results are really in the adult stem cells, all right? And that totally obviates any bioethical issues because now we can obtain consent and so forth, and we're not killing the human embryos. Okay, moving forward, anyone heard of Yamanaka? Yeah, Okay, what do you do? Shinji Yamanaka.
He did research with the stem cells.
Yeah, and he managed to do something that nature never does.
Induce.
Exactly, the IPS, okay? Induce pluripotent. In other words, he took cells that were fully differentiated, okay, fibroblasts. He took human fibroblasts, come from the mesoderm, and by discovering some factors, what are factors? What are these factors? These are proteins, these are regulatory proteins in the early development, in the early stage of differentiation of the cells, okay? They're regulatory proteins. Remember, factors typically are proteins. So I think it came down to about 4 proteins that are the regulatory proteins that establish, that regulate differentiation. And so when these proteins are added to the cells, these cells undifferentiate or de-differentiate, all right? They go backwards to a previous stage, all right? As they go, they go through multipotent all the way down to pluripotent, but not totipotent, because he did not want to generate embryos, okay? He did not want to generate zygotes or morula. He wanted to generate pluripotent stem cells because they are the gold standard, right? They're the theoretical gold standard. So he took fully differentiated cells, fibroblasts, undifferentiated them by adding these factors in particular proportions, or actually targeting them into the cells. And those cells de-differentiated, and then redifferentiated into a different tissue. So he was able to shift from one tissue, from fibroblasts, into differentiated tissues, in his case, different types of muscle, the 3 types of muscle tissues, skeletal, smooth, or the cardiac muscle, all right? Now, all these skeletal, smooth, and cardiac are fully differentiated cells. Sometimes some of them are multinucleate, like the skeletal, but at any rate, they are fully differentiated. So he did this, which is, he reverted them back to the pluripotent stage, not all the way to totipotent, right? So he stopped short. But there's a little I that stands for what? Induced, meaning that this is not done by nature. This is not natural, okay? It's induced, it's done in the lab, but it can be done. And he got these redifferentiated cells that were functional, all right? Or at least they were expressing, functional meaning that at least in vitro they would express the factors that they normally express in vivo, all right? So this was astonishing, was spectacular. And I was actually at Purdue. I was doing my PhD in genetics at that time in Purdue when this came up, and because I was looking at structure and function of DNA, this was very exciting research. And I was reading, he was interviewed, and I was reading the interviews of how he was under pressure. He was working sometimes 18, 20 hours a day with his team to get this done, because several labs in the world were trying to do this at the same time. Similar to what happened when Watson and Crick deciphered the genetic code. There are several labs in the world that were trying to do the same thing, you know, in England and States and other parts. So it's a lot of competition, and he got it done with his team. The motivating factor was very interesting to me, because he was working originally with embryonic stem cells, with human embryonic stem cells, and he was disaggregating human embryos. And at one point, looking under the scope at the human embryonic, the human embryos, the human blastocysts that he was disaggregating, he thought to himself, and he commented this in the interview. I don't know if you ever read those interviews. He actually commented, stick with me because this was 2002. I was there from 2001 to 2005. So we're talking about, you know, at least 50 years ago. He said, he started thinking to himself, I think he had several daughters. He says, these human blastocysts look very much like my daughters looked like when they were that stage. That was his motivation for looking for an alternative to find pluripotent cells without having to destroy human embryos. Because he said that, and I was so taken by it. He says, He was so honest. In other words, this is what I mean by professional integrity, scientific integrity. This is a man who knew what his daughters look like. In other words, he knew what the human embryo looks like at that stage of development. Looks exactly like a blastocyst. Okay? Doesn't look like us born because they are not born, but they look exactly like that every single time. So he was granting the humanity, he was impressed by the humanity, and he realized that he was actually killing human embryos. Yeah, and he says, I don't want to do this. That motivated him, and stellar success. So he'll go down in history, medical history, for being the one to invent IPS, which is being used today in a lot of research. So there we have it. Other sources. It turns out that the umbilical cord cells, okay, umbilical cord cells are also good to be used, and they are bioethically non-controversial. They're known for short as cord cells, all right, cord cells. But they come actually from the umbilical cord blood. So what do we mean by umbilical cord blood? Here's baby, baby's born, baby's attached to what? To a placenta, through a tube, the umbilical cord, right? And inside the umbilical cord, there is blood. So that's known as cord blood, all right, cord blood. And inside that cord blood, there are stem cells, all right? Where those stem cells come from? Junior, the baby, right? So they are embryonic-like. In other words, they're pristine, they're fresh, they haven't been exposed to much radiation or chemicals or anything. They're very tender, pristine, all right? And they are technically adult stem cells because the baby is already born, but they tend to have more of an embryonic quality in the sense that they are so tender and pristine and healthy because of no exposure essentially to what the rest of us are exposed over the years, okay? And so those are known essentially as mesenchymal stem cells, mesenchymal, so MSC is the other acronym here. Mesenchymal from mesenchyma. And mesenchymal stem cells can generate liver, fat, cardiac cells, nerve cells, cartilage, muscle, all right? So a variety of cells, tissues can be generated from these mesenchymal stem cells. They are from inside the umbilical cord. They can be saved. Junior is not harmed at all because this cord normally is what? When the baby is born, throw away. It's medical waste, right? So the cord and the placenta, normally medical waste. Now the placenta is being used for some research, and they're discovering even in the placenta some stem cells. Okay? But it's mostly the cord blood. So the cord blood has to be drawn as soon as the baby is born, all right? The blood is drawn and spun down, and the cells isolated. And the stem cells that are isolated, then what is done with those stem cells? They're not going to be used right away. Let's say baby's born normal. They're stored, right? Cryopreserved. We cryopreserve them. And of course, the hope is that they will never be used. But maybe 20, 40, 60 years down the line, that baby gets some injury or trauma or disease. Well, he or she has banked stem cells. And they're autologous, so there's no immune rejection, et cetera, et cetera. Maybe this junior doesn't need them, but maybe one of his siblings does need them. There's a good match there. Maybe the parents need them at some point, all right? And so they could be good for the immediate family because there's going to be a good match. In general, there's going to be a good immuno match, right? Because they have some degree of compatibility with the parents for sure, at least 50%, with the siblings, 25% in principle. So they're good. What's the hitch? What's the glitch with the umbilical cord blood cells, or cord stem cells, as they're known for short, cord stem cells? So far, insurance doesn't cover it. Therefore, out of pocket. And typically, the couple's having children, they're a young couple, they're getting started, they don't have that much money to spare. The procedure itself, it's about anywhere from $1,000 to $3,000, depending on where it's done, how it's done. You can buy the kit online, actually, and then you have to prep your people. You have to tell your doctor that you want to do this, and you have to tell the nurse, and at least the doctor will tell the nurse, and get the actual kit to the nurse so that the blood will be drawn as the baby's born, right, before they discard the umbilical cord. So that can cost maybe $1,000 to $2,000, get the kit and have it drawn, actually, which is overpriced, because what does it take for a syringe to draw the blood? All right, but it's a markup because they have to pay the overhead of a hospital or a clinic setting. And then there is the processing through the lab and the storage. And the storage, I hear, could be several $100s a month, maybe, or maybe a year, $200, $300. Anyway, what happens is that it starts adding up over time, and year after year. Sometimes couples can afford it. I try to promote it for couples to get their insurance company to pitch in something, at least something, because this can be seen as preventive medicine, in the sense that if these cells are good, they're autologous and so forth, they should bring down the cost of medicine in the population. The more umbilical cord banks, there are umbilical cord banks now, that there are banks that are with registry and all that, all right? So in the long run, this could lower the cost of medicine, and therefore insurance. And I'm trying to find an incentive for insurance companies to pay for at least part of this procedure, that it will be affordable for a larger number of couples having their babies. But it's a very viable option. There's no ill health to the baby or anything, and so would they be ethically controversial or not? Ethically good or bad? Good, right? Because the baby is not harmed at all. This is on the umbilical cord. All right? So when you hear about cord blood, and yet there's a lot of misunderstanding and misconception and misinformation. It's amazing, the ignorance. I remember when I was getting ready to go to Purdue, I spent a year in Boston. I think I told you I was preparing, reviewing the whole biology program and all that, because I've been 30 years since I've taken GREs and everything else. So I was doing research in a hospital in Boston. It's a large hospital, St. Elizabeth Medical Center. It's a research hospital because it's a research hospital for the medical school, for Tufts University Medical School. And so they have a whole separate building for research, in a lot of hematopoietic research being done there. And those labs, that research building, also did all the lab work for the hospital. So there was that advantage. At any rate, I was just doing volunteer research there one time, and someone told me an anecdote when we were talking about this umbilical cord cells, that they heard a nurse in the Catholic hospital, all right, where a couple came up, they were having their first child, and said, look, we want to save the cord blood because we heard about these mesenchymal cells and so forth, and they were talking to the nurse about it. And you know what the nurse said? Says, oh, I'm sorry, we don't do that here because this is a Catholic hospital. Like implying, stem cell research that has to do with killing human embryos. This is a nurse in a Catholic hospital. And she said, no, we don't do this because it's a Catholic hospital. And this is exactly the good thing they want to do, Yeah. And this is a research hospital for a medical school. So that's the level of ignorance. It's not just in the general population, it's even within the profession. And so you have a lot to do out there. When you get out there with bioethics, you're going to find a lot of ignorance, people who just don't know, but they have an opinion. And that's very dangerous, because they don't know, but they have an opinion. This nurse was convinced that this was a no-no, that this was a taboo. Okay. So, with regards to the stem cell issue and field, basically, you have to make distinctions at two levels. Once you bring it down to the level of human, then either adult or embryonic. And within the adult, you have straight up adult stem cells that we all have in our bodies, or you have the IPS, which are done in the lab, or you have the cord blood stem cells as another alternative, but so far they're kind of pricey still to maintain, all right, to obtain and maintain. Questions, comments about stem cell and the research? It's a promising field, it's increasing, there's a lot of money going into it, and it's another avenue that is exciting, you know, promising as we get more into regenerative medicine and personalized medicine. That is a big push for that today, okay? All right, then we have a couple minutes just for the vaccines. What's involved here is the following. Okay, so not all vaccines are controversial, but some are from the biological perspective because they were derived from aborted fetal tissues, okay? Now this whole area of vaccines started in the 60s, big time, to try to prevent epidemics and outbreaks of contamination with viruses mostly, because, you know, antibiotics work down to the level of bacteria, right? And antifungal will also work with fungi. But when we get down to the level of the virus, what is essentially the only thing that kills a virus naturally?
Or antibodies?
Our immune system, antibodies, okay, our immune system. And so with a virus, if we were to give an antidote of a person infected with a virus, that antidote is so strong that it would actually kill the person, kill the patient, all right? So we depend on the immune system. And that's what vaccines do, really. They fake the infection one way or another. It's either a killed virus or an attenuated virus, all right, a fragment of the virus or something like that, that will stimulate the immune system to react. But it's attenuated, it's toned down so that the person, and that's why vaccines work in the population 99.9% of the time, right? Or 99.9% of the population. What does that mean? Is, I mean, 0.1% of the population is gonna get the disease of the virus, whatever it is. Now, it could be, we could be talking about a flu that can be, you know, managed, or we could talk about rubella or mumps or rabies, something serious, okay? And so that's vaccines statistically will never be 100% because in practice, functionally, we're talking about real life human populations and they're all finite numbers, all right? So that's the drawback of the vaccine. But the good thing about it is that for the vast majority of the population, it will work. It will work. And some people will have some degree, there will be ranges of reaction, but in the vast majority of the population will be a minor reaction. And the positive thing is developing that immune resistance to the virus. So vaccines are developed all the time. They take a long time to develop. It costs millions of dollars, and they need tissue to develop them. So back in the 80s, I think it was, somewhere between the 60s and the 80s, some cell strains that were used for developing vaccines for humans, those strains came from aborted fetuses, okay? One was a lung tissue, and the other one was, let's see, it was rubella. I forget what tissue they used, but they used some tissue from a procured abortion from a woman who had an abortion, a voluntary procured abortion, all right, and then that tissue was used, the tissue from that fetus was used to develop these cell strains. And the vaccines were developed on those strains. So that is what we call ethically tainted ethically tainted cell strains, all right? In other words, that they were derived, those cells came from a source that was unethical. It was a procured abortion. Now, the pharmaceutical companies that developed these developed these vaccines were not involved in the actual abortion, but they used the tissue from that abortion, all right, from that fetus. So that makes them complicit, cooperating, enabling that tissue to be used for something. And so it's the whole area, the field of cooperation is known as cooperation with evil in bioethics. And there are some limits of that cooperation. Some of it is allowed, but some of it is not allowed. It has to do with the will engaging or not engaging. I'll get to that part. But before I get there, again, covering first the biology of it and then the bioethics, over time, from these cell strains that came from these aborted fetuses, all of these vaccines have been developed. The first one was rubella, and then the MMR, which is a triple, measles, mumps, and rubella, all right? And then varicella, which is also known as chickenpox, varicella en español. One type of hepatitis, zoster, which is also a type of shingles, adenovirus infection, and rabies. So all of these vaccines have been developed. Different, it also depends on the pharmaceutical company. It's more detailed, actually. It depends on the pharmaceutical company and so forth. But all of these, some are very popular, others are not distributed, are not given regularly, you know, like rabies and adenovirus, shingles more often, hepatitis A less. But certainly the MMR and chickenpox and shingles are given more commonly, all right, and particularly in children. So here comes the issue of consent, the proxy consent for the children. What do parents do, especially parents who are pro-life and they don't want to be involved in any way, shape, or form with a procured abortion? They don't want to be cooperating in that act, all right? And by using, so can parents who are pro-life and don't want to have anything to do with a procured abortion, can they use ethically these vaccines on their children, for example, or even on themselves, you know, when there's an issue of public health involved here? What degree of cooperation is involved, okay? Because they were derived, these vaccines were derived from these aborted fetuses, okay? So that's the controversial part. And believe me, for the general public, sometimes this could be a real issue. Why? Because there's another level of complexity here, since a number of these, especially the MMR, maybe the chickenpox, for children has to do with school. The children will not be allowed in school unless they are vaccinated. So it's by law now. What do parents do? Because you have conscientious objection, you know, the freedom of conscience to object, to not be forced to participate in an act that they considered highly offensive. And at the same time, they need to put their kids in school because they have a right, first of all, for the education of the child. And secondly, by law, the child has to go to elementary and secondary school. So what to do is a challenge.
Well, you can just write, some parents write notes though, like about it's against their religion. And
correct.
Like
and so, but it's state by state. Some states are more rigorous and some states are more lenient, all right? So it's a patchwork. It's not, there's no uniformity. And so if parents move from one state to another, what was legal in one state may be illegal in another state, not recognized. And even within the state, sometimes it goes down to county, especially in public schools, because the county, the public school system has a lot to say. They have to look after what? They have to look after the common good, all of the children, okay? And so there's a potential that a child that doesn't receive the immunization will contaminate the whole classroom. Because kids, the water fountain, they play with each other, they touch each other, you know, sometimes they eat each other's lunch, and so there's going to be contamination there. They're certainly coughing and sneezing on each other all the time. So. All right, so we have to, we can carve it out, all right? And it has to do with this principle of cooperation. And like I mentioned, it has to do with the will, if the will is engaged or not. So essentially, cooperation, it can be subdivided into formal or material cooperation. First of all, we have to understand, let me back up for a moment. Cooperation means that we're not the principal agent, okay? In other words, we're certainly not the people who procured the abortion. We're not the moms who had the voluntary abortion. We're not the clinic that did the abortion, all right? We're removed from that, so we're not the principal agents involved. But we, at one point, are we cooperating with that unethical activity, all right? We, at the level of consumer, we're not even the pharmaceutical company that is developing the vaccine. So you see, we're already 2 degrees detached from the offensive act itself. Not the clinic that performed the abortion or associated with anything that went there. We're not the pharmaceutical company that developed the vaccine. We're at the consumer end, all right? And so as long as the will is not engaged, in other words, as long as I did not will that offensive act, as long as I did not will the offensive act of the, that I was not in agreement with the procured abortion, then at most I am participating only materially. It's a material cooperation because I am using one of the products that came from that tissue, okay? So I'm using one of the products that came, so there's a level of cooperation, but it's minimal. It's only material cooperation, And so whereas formal cooperation with an evil act, with an intrinsically evil act, that's not licit. But material cooperation may be licit for a grave reason, okay? For a sufficiently grave or serious reason. For example, the health of my child and the health of the other children in the classroom of my child. That's certainly a grave, serious reason to immunize my child with any one of these vaccines, okay? And so looking at the particular good of my children and the common good of the other children, I can move forward because I certainly did not want to be associated. My will was not engaged in that. I did not desire the offensive act, So
I'm just gonna, I'm not against vaccines, but it's just playing devil's advocate real quick.
Yeah, good. Like that's what we need.
Okay, when we're talking about transplant, when the whole transplant of organs, it's clearly stated that you're not gonna, the reason you have to go through medical clearance is because you're not gonna risk the life of one person for the other.
Exactly.
But in this case, even though you're not complicit with the actual abortion and your 3rd-degree cooperation with it, aren't you still, because you're the consumer of the product, if there was no consumer, then there would be no product because no one. So because there's a consumer, the product is being made and it's being derived from this aborted fetus. So you're kind of still illicit to the fact that the abortion is being taken place because, so isn't this kind of
one way or another, you're in the pipeline. We're in the pipeline on the consuming end, right? Okay, so that's a very valid argument. Therefore, what could be required? So the vaccine itself may be used, but also there may be some extra requirement, at least on the part of the adults or the parents, to write, for example, to the vaccine company and to put some economic pressure and even political pressure if possible to develop an alternative line. First of all, this assumes, of course, that there's no alternative line, that there is not a line, there's not a vaccine that was developed from a clean strain, from a strain that is ethical, all right? But that does put on certain pressure on the part of parents to write to these pharmaceutical companies to develop what is known as ethically clean vaccines, all right? Or ethically licit vaccines. And in fact, that has been going on, and these parents have actually grouped because they're strengthening numbers and put enough pressure to these pharmaceutical companies that now the pharmaceutical companies are developing these new vaccines, even though it's extra cost, but they're making the millions anyway, to develop these vaccines that are ethically clean, all right? And they're using, look, it's as simple as using tissue that is not from a procured abortion. It could be from a spontaneous abortion, for example, where there's no immorality. You know, there's sufficient human tissue available that doesn't have to be that procured tissue, okay? That procured abortion. Originally, to the, not justification, but to the explanation of why this mess happened is because the doctor who developed the rubella vaccine actually was giving the mother who aborted, she had rubella. And rubella on a pregnant mom may cause defects on the baby, all right? And so she elected to abort her baby on purpose because she didn't want her child to have the consequences of the rubella. And so the child was already immunized, his tissue, the embryo was already immunized to have developed the antibodies. And that's why clinically it was ideal for this company to use that tissue, all right? Not justified, and they didn't think anything about, well, we didn't do the abortion, so why should we be blamed? But it has all these consequences. They didn't think enough farsight to see that for many people in our society, it would be offensive to participate in that. And that was originally the only rubella vaccine that was available. And then they did it, they took it up to the MMR, which is the triple, which is for children, you know. So it was a big faux pas, if you will, on the part of the pharmaceutical company not to have enough foresight. They thought that they were removed from the abortion issue because they didn't do the abortion, but they were using the tissue that was ethically tainted. And so parents did exactly what you said. They put enough pressure so that now these pharmaceutical companies are developing the alternative lines. And that then takes care of the issue, you know. But yeah, so there's an extra responsibility there. While the product may be used, there's that extra responsibility to make the statement, give us an alternative vaccine, you know, that is ethically clean. And so it's a happy ending because this has happened, and it's because of people that took the matter into their hands and spoke up and grouped together and put that pressure. Mm-hmm. Okay. Okay, so basically that's what I have. Oops. So if no questions or comments, no, people are good, then this takes care of Friday's lecture also. So we're not meeting on Friday? Yeah, because I was asked to do something else for theology right at this time, 11 and 12. So yeah, this is it. And I want to thank you very much for your attentiveness and wish you all the best in your studies and your professional pursuits, okay? Stay focused. I know it's a lot of work, but you get the hang of it after a while, all right? So this will be due by Wednesday midnight, as always. It will be the last grade, but you know, you have a very good track record so far, as you know, individually. If you have any questions or comments, just call me or email me, and we'll get it done. But to get that summary in by Wednesday, because then I have to put in grades. Okay. Anything else? No. If you haven't done your evaluation, I don't know who has and who has not, but if you haven't, please do it, because it's my way of making the course better. All right. Okay. Thanks again. All the best. Let's stop here the recording. Okay. Bye. See you.